Dear viewers, in the part of this program, today we will discuss about the phytohormones. Phytohormones are important chemicals which are present in the plant. So, at this time we will discuss about the occurrence, classification, importance and usage of these phytohormones in plant growth and development. So, first of all, what are the plant hormones? Actually, in plants, many behavioral patterns and functions are controlled by the hormones. These are chemical messengers influencing many patterns of plant growth and development, and they are known as plant hormones or phytohormones. Phyto means plant. So, we can say phytohormones or plant hormones. Even we can say plant growth regulators. So, the question is, the plants need the phytohormones for their growth and development, but some other chemicals, even water, fertilizers and manures are also being utilized by the plants for growth and development. Then which are the chemicals which we have identified as a growth hormones or phytohormones? So, in order to distinguish between phytohormones and other chemicals, these are the three basic requirements for being a hormone. That is, the synthesis of the hormone must be in one part of the organisms, and from there must be transport of the hormone to another part, and during this transport there should be induction of chemical response. So, for a plant hormone, these are the basic requirements, that is, it should be synthesized in the one part, should be transport to the another part where it should take a action. So, broadly if we discuss, the growth hormones or phytohormones have been classified into 5 groups, that is auxins, gibberellins, cytokinins, ethylene and abscisic acid. Among these, auxins, gibberellins and cytokinins are categorized as growth promoters, means their presence in the plant system accelerate the growth at various stages. The ethylene is called as a ripening hormone, because in the fruits, the ripening of the fruits is controlled by the presence of the ethylene. Abscisic acid has been categorized as a stress hormone, means the accumulation of abscisic acid increases when plants are under adverse conditions. In that case, the accumulation of auxins, gibberellins and cytokinin decreases. Now come to the auxins, the first discovered hormones. What are the auxins? Auxins broadly may be defined as an organic substance which promotes growth when applied in low concentration to shoot of the plants. It means, if we apply the auxins to the apical portion of the any part of the plant, the growth will be accelerated. Actually, Fritz Bent, a scientist from Holland, who in 1926 first discovered and used the term auxin. Auxin is a green word which means to increase, and he did this experiment in Avena coleoptile and found that when he put a small amount of auxin to the tip of the Avena, there was a curvature towards the light. So, how the auxins are present in the plant, where it occurs in the plants? The greatest concentration of auxin is usually found in growing apices of the plants, that is in the coleoptile tips, in the buds or in the growing tips of leaves and roots. However, auxins are found widely distributed throughout the plant body. It may be stated that where there is the active growth, there is presence of auxin. In 1934, Thyman and Skoog worked on Bissia fava seedlings which were grown in the light, and they have found that the concentration of auxin was present in the organs in this manner, that is maximum in apical buds, followed by young leaves and followed by mature leaves. These are the structure of some natural oxygens: IAA, indole acetic acid; IAN, indole 3 acetonitrile; IAC, indole 3 acetaldehyde; ethylene, indole acetate; indole 3 pyruvic acid and indole 3 ethanol. These are some of the oxygens which are naturally present in the plants. Among these, IAA is the most abundant, and whatever we are talking about the oxygens, basically we are talking about IAA, indole acetic acid. It is also possible to synthesize the oxygens artificially in the factories. 2,4-D is well known herbicide which is utilized to destroy the broad leaved weeds. This is basically an oxygen. When we apply 2,4-D in a small concentration, it accelerates the growth, but when we use in high concentration, it destroys the plants. These NAA and IBA are another synthetic oxygens which are utilized widely in the horticulture crops for rooting of cuttings. That we will discuss in the coming slides. Then what are the functions played by the oxygens? Cell elongation is the one of the most dramatic and rapid response induced by oxygen in isolated stem and calypyral sections. Whatever growth the plants are getting, they are the combinations of the oxygens and gibberellins. The apical dominance means the suppression of the emergence of lateral buds by the presence of shoot apex is called apical dominance, and by this process the oxygens controls the emergence of the lateral buds. This phenomena is very important in the flowering plants. You know, the plants, they always attract towards the light. The shoot portion of the plant has attraction towards light, whereas the root portion has the attraction towards the gravitational force. So, this phenomena is controlled by the oxygens in the plants. The roots are very important, and the oxygens stimulates root initiation on stem cuttings. Actually, in tissue culture, you have learned about tissue culture, the growing the plants in the artificial conditions. Then for tissue culture, the roots are initiated by artificial application of the oxygens. Similarly, flower initiation and fruit setting are also controlled by the oxygens. Oxygens are synthesized in the pollen and developing seeds and are known to involve in the flower initiation and regulation of fruit development. There are number of fruits like apple, guava, lychee, they all are controlled. Their flower initiation and fruit setting can also be controlled by artificial application of oxygens. Now, in agriculture, oxygens has various uses, like in germination. Some of the crops, they have the phenomena of seed dormancy, means their seed cannot be taken as such for germination. In that case, oxygen at very low concentration promotes germination, and however, its higher concentration prolongs seed treatment, retard or reduce the germination. So, for enhancing the germination, a low concentration of oxygens can be used. Rooting of cuttings in horticulture is very important, and oxygens play an important role in rooting of stem cuttings. Rooting is enhanced if the excised leaf or stem cutting is dipped in an oxygen solution, which increases the initiation of adventitious roots at the cut end. And in this case, IBA is the synthetic oxygen is most commonly used. Actually, this phenomena is widely utilized in floriculture and vegetable crops. For flowering, naphthalene acetic acid and 2,4-dihydroxy acetic acid, that is 2,4-D, are used to induce flowering in lychee, apple and pineapple. Actually, these crops, they have limited flowering in the beginning, or sometimes when there is the environmental variables, their flowering is affected. So, in order to optimize the flowering, these NAA and 2,4-D are used. The stimulation of fruit set. After flowering, fruit set is very important for getting the maximum yield in fruit crops. So, when environmental conditions are not suitable to fruit set, the application of oxygen to flower can promote this process, and in this case the concentration and time of the application of oxygen is very important, and that can be decided on the basis of the available literature and practical knowledge of the farmers as well as agriculture scientists. This is the prevention of premature fruit drop. Fruit drop in some of the crops is a phenomena which reduces the crop yield. For this, NAA, naphthalene acetic acid, is used at 10 to 20 ppm just prior to the beginning of the fruit crop in apple. In this way, we can reduce the fruit drop in apple. Similarly, in citrus, 2,4-D at 25 ppm can prevent the premature fruit drop. 2,4-D has also been found successful in preventing defoliation of cabbage and cauliflower. Thus, we can say that the application of NAA and 2,4-D can reduce premature fruit drop, increase the yield of the crops. Tissue culture and organ culture. Tissue culture is a technique to grow the plants under artificial conditions, where a combination of oxygen and cytokinin is very important to induce root and shoot portion. So, 2,4-D, NAA and IVA can be used in tissue culture. A favorable ratio of oxygen to cytokinin is very important to maintain root shoot ratio. The oxygens always initiate the roots and cytokinin initiate the roots. So, the concentration of oxygen can be utilized to initiate the roots in the tissue culture raised plants. Herbicide action. The 2,4-D is commonly used to control broad leaf weeds, dicot weeds in commercial cereal fields. The picloram is used for vegetation control on non-crop land because of its high activity and soil persistence. The 2,4-D, when we use in small concentration, it accelerates the growth. When we use in high concentration, it controls the broad leaf weeds. Now come to gibberellins, another important growth hormone. Gibberellins has been identified through a fungus Gibberella physicorae, and these may be defined as a compound which is active in gibberellin bioassays and possesses a gibberellin ring skeleton. There are many other compounds which are active in some of the assays, but do not possess a gibberellin ring. Such compounds have been called gibberellin like rather than gibberellins. These are the structures. Till date, a number of gibberellins have been found, and today the counting has reached to 125. Among these, GA3 is the most common, and whatever we are talking about the gibberellins, this is the GA3. So, the gibberellins were originally isolated from a fungus, but now they have been shown to be present in all the groups of plant kingdom, including angiosperms, gymnosperms, ferns, mosses and algae, but are unknown in bacteria. All the organs of the flowering plants contain gibberellins, but the highest level has been detected in seeds. Young leaves and roots are also rich in gibberellins. It may be generalized that rapidly growing and developing regions of the plant possess higher concentration of gibberellins. So, gibberellin is present almost in all parts of the plant, fern, mosses except in bacteria. These are the chemical structure of some of the gibberellins: GA3, GA4, GA7 and GA13. So, what are the important functions of gibberellins? Gibberellins are involved in the cell growth. The oxygens are involved in the cell elongation. The gibberellins promotes growth by affecting cell expansion or cell division. For example, internodes of tall peas have more cells than those of dwarf peas, and cells are longer. The bolting in long-lived plants. Actually, in many plants, gibberellins function as a controlling factor in a balance between internode growth and leaf development. For example, cabbage and other species in the roseate form that has short internodes sometimes grow 2 meter tall and then flower after GS application, whereas untreated plants remain short and vegetative. Means the application of gibberellin can enhance the flowering in the tall plants. Watermelon, squash and cucumber are also elongate faster when applied with the gibberellins. Gibberellins can also substitute the flowering, particularly in long day plants. They are also able to overcome some species need for an inductive cold period if they are to flower sooner. Actually, vernalization is needed in some of the plants, but the application of gibberellins can mitigate the effect of the vernalization in these plants. Seed dormancy, like gibberellic acid can overcome seed dormancy by acting as a substitute for environmental variables or red light. Actually, in the germination, red light is needed for the seeds, but application of gibberellin can substitute for this red light. GA also stimulate the production of numerous hydrolases like alpha amylase by the alluvial layer of germinating cereal grains. This property of gibberellins is utilized commercially in the brewing industry in the production of malt, and you know in beer making the gibberellins are utilized in the barley. The male female ratio is very important, particularly in cucurbitaceae plants, and this gibberellins are utilized for the induction of maleness in dioecious flowers. For example, it promotes male flowering in cucumbers for seed production. Parthenocarpy is the production of the seedless fruits, and gibberellins play an important role in the parthenocarpy. Gibberellins, especially GA4 and GA7, have also been found reliable in producing parthenocarpic fruit set. For example, palm and stone fruits are generally unresponsive to oxygen treatment, but gibberellins can induce parthenocarpy in these fruits. Production of seedless fruits in grapes in an example which are produced by the application of the gibberellins. The third group of the phytohormone is the cytokinin. Cytokinins means the growth promoters involved in the cell division. Actually, in 1965, Skoog, Strong and Miller identified a chemical which, regardless of their activities, promotes cytokinesis, means cell division in cells of various plant organs. And Fox again defined cytokinins as chemicals composed of one hydrophilic adenine group of high specificity and one lipophilic group without specificity. Fruits and endosperm are the richest source of cytokinins. Coconut milk and corn endosperm possesses the active substances. Substances with cytokinin activity have also been reported in tomato juice, in floral extract of apples and pears. The cytokinin-like substance is also present in peach embryos and sunflower root exudates. These are the chemical structures of the cytokinins, that is G18 and 6-benzyladenine is the most abundant cytokinin. Now, what are the functions of cytokinins? The major function of cytokinin is the cell division. The exogenous application of cytokinins induces cell division in tissue culture in the presence of oxygens. This also occurs endogenously in crown gall tumors on plants. The cytokinins play important role in delaying the leaf senescence. The Richmond and Long, they have proved in the Gentium that cytokinin was able to postpone the disappearance of chlorophyll for several days, which normally occur with the aging process of leaves. So, it delays the senescence in most of the green parts of the plants. The source sink activity is very important, and cytokinin enhances the sink activity in most of the plants including wheat. This phenomena is known as the cytokinin induced nutrient mobilization. The stomata, which are the entry point in the plants, their activity is governed by the cytokinin. When cytokinin is applied to isolated epidermis of a grass, it causes stomatal opening. Now, we come to another growth hormone, that is abscisic acid. We call it as a stress hormone. The abscisic acid in the plants is present in the roots, stems, buds, leaves, means every part of the plants, and chloroplasts are in the leaves contain the carotenoids from which AB arises. The like gibberellins and cytokinins, the abscisic acid moves monopolarly. There are no synthetic abscisic acid, means we cannot synthesize abscisic acid in the factories. And this is the chemical structure of this abscisic acid, and the major functions governed by the abscisic acid is the stomatal opening and closing. Whenever I told you that whenever it is called a stress hormones, means if plants are under any stress conditions, they will close the stomata by accumulating high concentration of the abscisic acid. The ABA also acts as a stress hormone. The level of ABA increases when plant is exposed to salt, cold or any type of the stress. It is suggested that ABA synthesis is regulated at the transcription level, and increasing level of ABA causes modification in gene expression to tolerate stress conditions. The dormancy are also induced by ABA. The oxygen and cytokinin, they inhibit the dormancy, whereas ABA causes the dormancy. At molecular level, it has been designated as quantitative trait that are controlled by combined action of several genes which are governed by the abscisic acid. So, in the part of this program, today we have discussed about the phytohormones, different classes of phytohormones, how they have discovered and how they are important for the growth and development of the plants. I told you about the oxygens, their practical utility of the oxygens, cytokinin and gibberellins, how they play role in cell division, cell enlargement and cell growth. We have also discussed about the ethylene, why it is called ripening hormone and how it is important in ripening the fruits. In the last, I told you about the concentration and the time of application of various hormones. The importance of abscisic acid as the stress hormone has also been discussed. Hope you have enjoyed this lecture. Thank you.